All right, so yes, my name is Kevin Snyder, and we are introduced to the title of my talk. And at first, I'd like to start with a standard disclaimer that this presentation reflects my own views and should not be construed to represent FDA views or policies. And now to give some background, so sendig is the non-clinical toxicology version of SDTM. And it really wasn't a thing until FDA published the binding guidance in 2014 that required both clinical and non-clinical studies be submitted with standardized electronic study data where applicable. And so the current types of studies that can be modeled in SEND are single and repeat dose general toxicology studies, carcinogenicity studies, and respiratory and safety, cardiovascular safety pharmacology studies. And as of this past October, last month, we've received over 6,000 SEND data sets, and so and we're getting thousands, right, 2,000 or 3,000 each year now. So this has really created great new opportunities for the collaborative development of open source software solutions to facilitate cross-study analysis of toxicology study data. And the work that I'm going to talk about today is really part of a collaboration between FDA, BioCelerate, and PHUSE. So BioCelerate is a subsidiary of TransCelerate. It's kind of the non-clinical TransCelerate, where some pharmaceutical companies have banded together to create a database of shared SEND data sets, and they're trying to obviously extract value from that database. And PHUSE is a kind of a CDISC implementation user group. And so we're all working together, and BioCelerate has agreed that we're going to publish the methods that we developed to extract value from these data sets. And so the first publication from this collaboration focused more on identifying challenges and opportunities, really identifying challenges, kind of where in the standard and the way it's being implemented are we going to hit roadblocks when we try to do cross-study analysis. And so we published a paper on that, and that's led to some good work, both in creating best practices, which we're publicizing through PHUSE, as well as some modification or some feedback to CDISC in how to kind of modify the actual standard itself to solve some of these problems down the road. For the second publication we produced, we actually explored a real use case. We decided to perform a historical control analysis of a database of SEND data sets, and we published the results of that. And I'm going to talk about how that has kind of led into the development of an R package. So for this project, we started out with a question. We wanted to, for example, just ask what are the most common microscopic or histopathological findings in male beagle dogs, 12 to 18 months of age, that were dosed by oral gavage. And what we found is that in order to ask this question, we obviously had to break it down into different components. Some would help us figure out which studies to include, some which animals to include in the analysis, and some, which specific records from the data we needed to pull. And with a little bit of work, we were able to kind of break each of these query blocks, we were calling them, into different R scripts or R functions, and kind of string them together and be able to perform the analysis of this question after pulling the SEND data into a SQLite database. And so we published this analysis and these scripts, with that paper, but it's really not something that other people can pick up and use. I don't know if you've ever tried to use someone else's scripts. It can be very messy, and also the data set we use isn't publicly available, and so that's another problem. And so this led us to think, well, let's take this a step further and actually refactor these scripts and create a real R package that will allow us to do this kind of work in the future, and also potentially allow others in the community to also utilize the work that we've done. So right now, the functionality of the package is focused mostly on this use case of historical control data analysis. However, we do plan to add additional functionality as we explore other use cases. We'll add functions to the package that will facilitate those use cases, such as comparing, contrasting toxicological profiles, or looking for off-target effects, things like that. And so all the source code for that original manuscript and the R package is available on the PHUSE GitHub repository under the BioCelerate project. And now I'm going to give some schematic diagrams showing how the package works. So the first set of functions, first thing you need to do is use a set of functions we built to actually build or maintain your database. And so this, we set it up so that it'll allow you to build a SQLite database. We chose SQLite because it's very lightweight. You don't need a server. It's pretty easy to set up on your desktop. If you just have a bunch of send data sets, you can build a database and start querying it. Although, as I'll talk about in a second, we did allow our query functions to connect to Oracle database, and we may put connectors to other kinds of databases as well. And so the workflow here, you create an empty database, create a schema, you can import your studies, index them, and then these workflows are just to add and delete studies. Then probably the more interesting thing you can do, now you can query your database once you have it. Like I said, this will work for SQLite and Oracle. As long as the Oracle database is just a straight upload of the send data with no changes of variable names or anything, as long as it's structured the same way that we've essentially structured the SQLite. And so this may look familiar. Like I said, we kind of adapted our scripts, and we got so we put different functions in that you can string together to perform historical control analysis. And like I said, we'll continue to add more functions and more functionality for other kinds of cross-study analysis in the future. And then the last thing we did, in order to make this work accessible to people who maybe aren't as familiar with R programming, we decided to wrap it in a Shiny app. And so we have a public version of this app running on shinyapps.io, and this is just running on a small database built from publicly available mock data sets. So it's not useful. I wouldn't use it to actually as reference ranges. I think there's only 5 studies in it, but it's useful in terms of demonstrating the capabilities of this tool and the package, and then you'd have to set it up in your own environment to run it on your own data. But briefly, I want to just go through how our Shiny app works, what functionality it can deliver. So it's set up as a dashboard where on the left, you have your filters. So you can select the date range, study design, route of administration, species, strain, sex that you want to include in your query. And then it'll generate line listings describing the characteristics of the animals that it pulled. And then you can look at historical control distributions for endpoints in different domains. Right now, we just have set it up for histopathology, the MI domain, clinical pathology, the LB domain, and body weights, the BW domain. So now I'm just going to give a couple of screenshots of just showing what the tool delivers in terms of results. And so this is the table for describing the metadata for the animals that you selected from your query. So you can kind of see which studies they're from, how old they were at the start of study, stuff like that. And then as you go through the domain tabs at the top, you can look at the histopath findings. So here is a filterable table. We filtered for kidney. You see what are the background rates of different findings in the kidney. In the LB domain, now I've filtered it to show the liver function tests. And so you can see what's the mean and standard deviation, kind of the reference ranges for control animals for different clinical pathology lab tests. And then body weights, we set it up so it calculates the age of the animal and then how much the animals in your query weighed at that age. And so you can basically generate growth curves for different strains and species and kind of know what the average age or average weight would be for different animals at different points in time. So in conclusion, the sendig package, it's free open source software solution that will enable data scientists and toxicologists to interrogate large repositories of send-formatted toxicology study data. And the current functionality focuses on historical control, but we'll plan to expand that to other use cases. And lastly, this is a collaborative project, so please feel free to try it out and reach out to me if you have questions about how to use it, or especially if you're interested in contributing. This has been a really fun collaboration to be a part of, and we're really making some good progress. We plan to publish to CRAN this winter, and also try to publish a accompanying manuscript in a journal just describing to publicize the work we've done. And last, I just want to acknowledge everyone who's contributed to this project, Jesse Anderson and Yusuf from FDA, Dan Russo, he was a previous fellow at FDA. He's now at Rutgers. Michael Rosentreiter from Bayer. And then our BioCelerate partners, Mark and Brianna from Lilly, Bo Larson from Novo Nordisk. Special shout out to Bo Larson. He is a great professional statistical programmer and really wrote the code for most of the package. He'll be leaving the project now. Wenshin Wang from BMS is replacing him in that role. And Bill Hauser also contributed enormously to this project and to PHUSE non-clinical efforts in general. So thank you, everyone. And